All right, the next section is going to look at the structure of DNA specifically. So again, chapter 12.1 is talking about the history of the discovery of DNA and all the scientists there. Again, I really don't want you to know all those scientists. I want you to more focus on this content. 12.2 is going to start with nucleotides, and it's going to move on to the structure of DNA and how it relates to its function. All right. So the first thing we're going to talk about here is nucleotides. Again, nucleotides are the building blocks of DNA and RNA and just all nucleic acid. However, nucleotides have three parts. They are the phosphate group, the ring-shaped sugar molecule deoxyribose, and the nitrogen-containing base. It's either a single ring or a double ring, and we'll talk about those a little bit later. You can see here this is a perfect example of a nucleotide. We have that phosphate group, we have that sugar molecule, and we have that cytosine, which is the base. So nitrogen-containing base or nitrogen nitrogenous base is just a molecule that's going to have nitrogen in it. So just remember that these guys are going to have nitrogen. And if you remember back from chapter 3, this is when we did the macromolecules. We talked about the different elements that were in each macromolecule. Nitrogen being one of those that's very specific to DNA. So again, nucleotides, like I just mentioned, are the monomers of DNA, and again, these are this is like the wall, and these are like the bricks that make up the wall. Now, there are four different types of nucleotides. We have adenine, guanine, thymine, and cytosine. If I ever, please correct me if I ever say cysteine. Cysteine is not a DNA molecule. I always just get the words mixed up there. Cytosine is a nucleotide. Cysteine is a amino acid. So they are somewhat kind of close in like this chapter, but I'm usually pretty good on on that. Just once in a while, I'll say cysteine. I'm like, wait, no, it's cytosine. So cytosine here, and you can see that they have different structures. You can see adenine, guanine look very similar, and cytosine and thymine look very similar. A's and T's we're going to learn that are going to bond together, and G's and C's are going to bond together. And you can see how these two have that double ring structure. We have one ring right here and one ring right there. Now, again, you can see a little bit difference in the amount of cytosine, thymine, guanine, adenine within each of these species. So it does differ. And there is some similarity between like us and chimpanzees just because we have such close common ancestors. But it's there's no really rhyme or reason that one has more of the other. It's just how they evolved. Now, Chargaff actually did work with Watson and Crick a little bit, so he was around that time period, and he discovered that the A's approximately equal to T's, G's approximately equal to C's. That means that A's, whenever you see an A, on the other side, you're going to see a T. G's, whenever you see a G, you're going to see a C on the other side, and that kind of makes sense. Like G's and C's bond, A's and T's bond. Again, I'm going to say like when they found this out, they're like, "Oh, this is so cool! Like this is revolutionary." This is like, I know you're sitting there like, wow, you're telling me that A's bond with T's. Like I learned this in middle school. Like, but this is during that time period. This was like huge news. So again, just like I said, A's bond with T's, G's bond with C's. However, we have to go a little bit another step deeper. Pyrimidines are single ring structures. That's cytosine and thymine. And I always tell kids to remember, hey, pyrimidines have Y's, so do cytosine and thymine. Now, we will get into uracil a little bit later, but not yet. Just remember they are pyrimidines. Purines, I always just remember pure as the with the U there has the two like just I just remember that whenever I say U, I see two. So adenine and guanine, and they do not have the Y's. And you can see their structure. We have a single ring here for the pyrimidines, and the purines have the double ring. So a purine has to bond with a pyrimidine. So let's say if one organism has 10,000 purines, they're going to have 10,000 pyrimidines. You can see structurally what they look like. You can see that they have the nitrogen here, and then you can see the adenine and guanine, the purines. They have the double ring structure, and the pyrimidines are the cytosine and thymine. Oh, so when we do these, it says you find a new bacteria and you sequence the genome and find the thymine percent is 21. What percent is adenine? Obviously 21. That's an easy question. But whenever you do it the other way, so again, let's say we have a geneticist sequences the genome of a lion and finds percent of guanine is 34. What's the percent of adenine? Those don't go together, but we can still find it out. You just got to take 34 and 34, add them together at 68. What's remaining after you subtract it from 100? 32, you can see 8T, so then we just divide that by 2, so we have adenine, which is 16. So it's just understanding that GC plus A and T equals 100%, and if you just do the math there, it's kind of simple, and I will go over this pretty quickly in class again, but it's pretty simple math just to figure out what adenine and thymine are. All right, finish out this section. DNA is going to consist of these nucleotides in a twisted ladder form, and if I have my thing, I'm just going to go real quick. These this twisted ladder, if you look at this model, and hopefully I'll use this in class, you can see this model is a great representation of DNA. We have two strands of it, and again, you can see how this twisted ladder looks like the one on your screen. This twisted ladder, again, you can see that it has two sides to it, looks like a ladder. The double helix refers to that there are two sides to this, and it is twisted. It's called a helical form. So you can see it's a twisted looking ladder. I love that representation on the right there that you see. However, it's kind of confusing to see how those things bond and how the makeup of nucleotides along the strand there. So I like this image. I think it's a lot better and a lot easier to kind of digest. If you look here, nucleotides have to be nucleotides have to be connected by covalent bonds. That's between the sugar and phosphates. And you can see here on this section, we have the sugar phosphate backbone. Now, if you remember back from chapter 2, which is chemistry, covalent bonds are very, very strong. So this backbone is very, very strong. And you can see these adenines and thymines bonding together, G's and C's, A's and T's. Between the nucleotides in the middle are these hydrogen bonds. We're going to learn that for every A and T, there's going to be 2 hydrogen bonds, and between G's and C's, there's going to be 3. Now, if you remember back again to chapter 2, hydrogen bonds are pretty weak. They're much, much weaker than these covalent bonds. So yes, we are going to be able to open this up, and it makes sense because we want to read this information. Every turn of the DNA has 10 nucleotides. Again, the sides, these are the sugar phosphate groups, and they're held together with covalent bonds. Like I said, complementary base pairing, A's and T's, G's and C's. There we go. We're good there. Hydrogen bonds each are between G's and C's, A's and T's. There's 3 between G and C and 2 between A and T. Another good meme. I'm sorry, G, but we're just basically not a good match. Basically, because of the bases. Okay, bad. All right, so you can see here, now this shows 3 different diagrams of this DNA molecule. You can see how this is the ribbon model. I like this model because it's showing you those curves. There's actually grooves that we're going to talk about next. I also like this one because it also shows you, it's starting to show you that the DNA is something called anti-parallel, where you have strands going in opposite directions. You can see how I always like to call the sugars like the houses, and they're facing up here, and they're facing down here. And then this is just the computer-generated model of the atoms. Let's talk about that anti-parallel. So you can see here, they're going to run in opposite directions, where we have this phosphate group here at the top, and at the bottom here we have the sugar. We call the phosphate group the 5' end, so I always like to say it's the 5' phosphate. So just like phenotype and physical characteristic, 5' phosphate. And then the 3' is the sugar. And you're going to ask, why do we call it 3 and 5? It deals with the number of carbon atoms and where they're at in the sugar. You can see here on that 5th sugar, we have the phosphate group. And on the 3rd, down here on the 3rd carbon, that's where you're going to have it. That's an opening for the next nucleotide to bond. So that's why they call it actually 3 and 5, but you don't have to know that. On the opposite side, it runs the opposite direction. So you can see here we have 3 to 5, and it looks like it's going in the opposite direction. Yeah. So these are anti-parallel, which is going to be really important going forward when we talk about replication, transcription, translation. I think that's the end for this chapter. Take a look. Oh, no, we have one more. Grooves within the DNA. I had this a couple years ago. It's not in your book, but I still think you should kind of know about it. There are major and minor grooves. These major grooves here are just like the larger locations, and it deals with how the DNA twists. I'm going to bring my screen back up here so you can see. This is actually not a 100% accurate model. If you look, there are no major and minor grooves. It's really hard to create those in models because it deals with how the model has to twist, and I would not be able to twist it just as easy as I do right now. So there are, like, you can see how both grooves, this one and this one, they're the same length. In the one that you see in your image, the major groove is much larger and the minor groove is much smaller. And it just deals with how the nucleotides are going to join together. All right, so this ends section 12.2. This is where the structure of DNA ends. The next thing we're going to go over is replication.